I'd like to first thank the organizer for inviting me. Just really glad to be back in person meeting again. So let me try to advance my slides. No. Okay. All right, so as the chair mentioned, you can use this QR code or the link to ask question, and it will be answered at the end. So I think, as many of you already know, child bone growth happens in the growth plate near the ends of a long bone. And I think it would be just appropriate just to give a very brief introduction to the growth plate to start our symposium. So the growth plate is composed of mainly chondrocytes that are arranged in, oops, can I go back? I'm just gonna talk. Arranged in three histologically distinct zones: the resting, proliferative, and the hypertrophic zone. So growth plate in the resting zones are stem cells that can self-renew and then give rise to these proliferative chondrocytes, which are then arranged in columns parallel to the long axis of the bone. And then in these columns, these cells divide rapidly, and as they do so, they gradually move down the column. And as they go near the bottom of the column, they stop dividing and start undergoing hypertrophy. This process of chondrocyte proliferation followed by hypertrophy lead to formation of new cartilage tissue, which are then replaced by bone tissue because of osteoblasts coming from below, or these chondrocytes transdifferentiate into osteoblasts. And so either way, this process leads to formation of new bones, and so there you go, longitudinal bone growth. And my talk is going to focus on discussing the epigenetic mechanism that regulate this process in the growth plate. So I think because this talk is about epigenetics, it's important to first try to define what epigenetics is for the purpose of my talk. So by definition, epigenetic modifications are heritable phenotypic changes that doesn't involve changing the underlying DNA sequence. In the literature, it's usually used to describe changes in chromatin structure, histone modifications, or DNA methylation. But in some broader definition, it could also include microRNA, long non-coding RNA, or transcription factor bindings, none of which involve changing the DNA sequence. But for the purpose of this talk, I would like to use the more narrow definition and try to just focus on the first three items. So let's talk about histones. So histones are, as you may know, spools that DNA wraps around and pack into nucleosomes in the cell. And so histone modification affect how tightly the DNA is wrapped around the histone, and therefore it affects how tightly affects the level of gene expression. Just to give you an example, you could have histone acetylation. Acetylation reduces the positive charge of these histones, and so weakening their interaction with the negatively charged DNA. And so histone acetylation usually leads to a more open chromatin structure, increased accessibility to transcription factor, and therefore an increase in gene expression. And then histone methylation has a more complicated effect on gene expression because histone can be monomethylated, dimethylated, or trimethylated, which means multiple methyl group being added. And then it can be modified at different places in the histone. And so depending on where it is, it could be associated with gene activation or repression. DNA methylation is, on the other hand, usually associated with gene repression, and it's done by DNA methyltransferases. There are different kinds of DNA methyltransferases. There's DNMT1, that's, sorry, to go back 2 slides. Next. There's DNMT1 that is responsible for genome imprinting, and then there's DNMT3 that is involved in de novo methylation. So when I talk about genome imprinting, you might be thinking about, these growth disorders like Backward-Wiedemann syndrome, Silver-Russell syndrome, that is caused by defect in genome imprinting, which is, a mechanism where DNA methylation control a group of genes called imprinted genes to be expressed or silenced, depending on whether you get the copy from your father or your mother. These imprinted genes, indeed, for example, IGF-2, are important regulators of child growth. And it probably deserves its own talk. And so I'm going to try to stay away from imprinted genes altogether and focus on mechanisms that more broadly control gene expression globally. So when I think about how one could study epigenetics of the growth plate, I feel like there could be, of course, many ways, but maybe three main approaches. First one would be to try to study the epigenetic and transcriptome profile of growth plate chondrocytes. And thanks to sequencing advanced technology, this has become more and more feasible. And for example, in this recent paper by Andrew Varkam's group, they look at epigenetic changes as chondrocyte transition from proliferative zone to the hypertrophic zone. And they did that in the embryonic mouse growth plate. And then the second approach would be to use some reverse genetics. Over the years, there are many genes that were being discovered to be epigenetic regulators. Usually people classify them as writers, erasers, or readers of epigenetic marks. So meaning you either add, remove, or just read the epigenetic marks in the chromosome. So one could try to knock out these genes in the growth plate, for example, in the mouse, and then look at the skeletal phenotype, and that these indeed had led to many insights into what these genes could be doing in the growth plate. But I'm going to focus on a more clinically relevant approach, which is to look at human growth disorders that is caused by epigenetic mutations or defects in epigenetic regulators. I feel like one good example to start would be the histone deacetylase, HDAC4. So this is actually some work done by Cronenberg's group, this overall field. So HDAC4 haploinsufficiency causes a genetic growth disorder called brachydactyly mental retardation syndrome. The role of HDAC4 in the growth plate has been quite well studied. There's a knockout of HDAC4 that show growth retardation, atopic bone formation, and early hypertrophy. And the transgenic mouse overexpressing HDAC4 has the opposite skeletal phenotype with a lack of bone mineralization and delayed hypertrophy. Now, let's recall that HDAC4 is a histone deacetylase, and so it deacetylates the histone to likely repress gene expression. And HDAC4 represses a lot of genes, but most importantly, it represses genes like RUNX2 and MEF2Cs that are transcription factors important for hypertrophy. And so when you have a knockout, you got upregulation of RUNX2 and MEF2Cs in the growth plate, and so you got some early hypertrophy. On the other hand, too much HDAC4 delays the expression of these genes, therefore you get impaired or delayed hypertrophy. Now, interestingly, more recently, HDAC4 along with HDAC5 has been shown to be involved in mediating the effect of PTHRP, also called parathyroid hormone-related peptide, in the growth plate. So you may know that PTHRP is expressed primarily in the resting zone, and it diffuses across the growth plate, forming a gradient. And because PTHRP suppresses hypertrophy, this gradient also determines where hypertrophic zone can start in the growth plate. It's when PTHRP level drops below a threshold. And it turns out this effect of PTHRP is mediated through activation of HDAC4, which then determines where RUNX2 and MEF2C is expressed in the growth plate. And importantly, this helps explain why HDAC4 haploinsufficiency and PTHRP mutation share a quite similar skeletal phenotype where PTHRP mutation causes autosomal dominant brachydactyly type E. So in addition to HDAC4 and 5, there's only a handful of genes on this list that is directly causing growth disorders. There is the KMT2 that and KMT6 that could cause a Kabuki syndrome, which has some decrease or short stature. But there, quite interestingly, there's a few of these genes that cause skeletal overgrowth. Particularly, there are 3 of them, quite phenotypically similar overgrowth syndrome, caused by writers of methylations. There's the Soto syndrome that's caused by NSD1 mutation that affects H3K36 methylation. Weaver syndrome is caused by H3K27 methylation due to EZH2 mutation. And then Tegenbaum-Raman syndrome is caused by DNMT3 mutation. And of these few diseases, we focus on the Weaver syndrome and EZH2 because of a patient that came to us at the NIH a while back. And I'm going to tell you a little more about this story very briefly. So EZH2 is part of the PLC2 complex that regulate or catalyze H3K27 trimethylation, which is a repressive chromatin mark. And in 2012, it was first discovered that heterozygous missense mutation of EZH2 causes Weaver syndrome. Around that same time, there's a patient at the NIH that has overgrowth and advanced bone age. We performed exome sequencing and found mutations of EZH2, therefore confirming that he has Weaver syndrome. Now, these patients are tall, but they have normal levels of growth hormone, and so we were wondering if the effect of EZH2 were directly acting on the growth plate, and so we decided to study that. And the approach we took was trying to make catalyst-specific knockouts of EZH, not just 2, but 1 and 2 together because of some functional redundancy of the 2 EZH in the PLC2 complex. And the mouse, the double knockout mouse, have decreased growth, including decreased bone length very early on. When we look at the growth plate, they will have shorter growth plate, and there's decrease in both proliferation in the proliferative zone when we try to label the cells in the cell cycle, and then there's also decreased size in the hypertrophic cells, which means they can't fully undergo hypertrophy. So we try to look at the mechanism, and so we use laser capture microdissection to isolate these cells and then look at the transcriptome. Previous slide. And as you can imagine, because EZH2 catalyzes a repressive mark, the loss of EZH2 will lead to upregulation of a lot of genes or pathways. And one of the things that we found was like a cell cycle checkpoint. And so we hypothesized that maybe EZH2 controls cell cycle progression. In particular, we look at a genomic locus that contains two CDK inhibitors, CDK N2A and B, which these binds to CDK4 and 6 to block cell cycle progression. And what we found is that in chondrocytes, H3K27 trimethylation is normally enriched in the promoters of these CDK inhibitors, but this mark is lost in chondrocyte that doesn't contain EZH2, the knockout chondrocytes, and that's associated with an increased expression of these inhibitors. We then confirmed that this effect is mediated through EZH2 because you can use, in chondrocyte, you can use an inhibitor to block EZH2 activity, and that will lead to upregulation of these inhibitors associated with a decrease in proliferation. And you can reverse that by using siRNA to block CDK inhibitors, which then partially rescue proliferation. So we then tried to do a similar study in the hypertrophic zone. Again, we found a lot of pathways that was changed in the knockout chondrocyte, one of which is Igf signaling. So Igf signaling is important for chondrocyte hypertrophy because, for example, in the knockout, the growth plate, particularly in the hypertrophic zone, the cells look smaller. And I'm just going to tell you what we found. EZH2 normally repress the expression of these proteins called IGF binding proteins. And these IGF binding proteins normally interact with the ligands to block the interaction with the receptor. And so the loss of EZH2 led to upregulation of these IGF binding proteins, therefore decreased IGF signaling. And so collectively, we found that EZH2 is important in the growth plate, both in the proliferative and the hypertrophic zone, by allowing cell cycle progression and also by promoting IGF signaling. And around the same time when we were studying the EZH mice, there was another group, actually Kobayashi's group, studying another mouse. That's the EED the EED knockout mouse. So EED is another member of the PRC2 complex. And very interestingly, the mouse have a very similar skeletal phenotype as our double knockout, except that they were focusing on Wnt signaling and TGF-beta signaling. And so we put together ours and their finding that would suggest the PRC2 complex is involved in many different pathways in the growth plate that are important for growth plate function. Now, there's one problem with this, and that is, that doesn't tell us very much about Weaver syndrome. These mice are all small instead of large. And so in order to understand that, we go back to look at Weaver syndrome. So let's recall that Weaver syndrome is caused by heterozygous missense mutation of EZH2 usually. And however, it wasn't so clear at that time whether these are gain of function or loss of function. And the fact that there's a lack of frame shift or early truncating mutation commonly found in in Weaver syndrome suggests that maybe it can't be just a simple loss of function. You can't just have loss of EZH2 and then just give you overgrowth. And because our knockout, which is a loss of function, have decreased growth, we wonder, could it be that you have increased activity of EZH2 that cause overgrowth? And so we decided to study EZH activity. Well, it turns out studying that is not very straightforward because EZH2 catalyze multiple steps in this reaction. And just using another disease as an example, in B-cell lymphoma that also is caused by EZH2 mutation, it was interesting that this mutation actually have a loss of activity in the first step, but a gain of activity in the second step in this reaction. And so if you just look at the mutation, it would look like a loss of function because it can't perform this first reaction. But if you look at it in a heterozygous state, because the wild type is present, it would be able to perform this first step, and then the second step is enhanced, so it will look like a gain of function. And so we just need to be aware of this. And so when we study the Weaver mutation, we first look at it in the knockout chondrocyte. That doesn't contain endogenous activity. In this setting, the Weaver mutation show an activity that's somewhere between the mock transfection, which is, has no activity, and the wild type, which has 100% activity, let's say. And so that would suggest a partial loss of function, but not a complete loss. And then when we look at the same thing in the wild type chondrocyte that contains endogenous activity, now the mutation just have similar activity as the wild type, perhaps because the wild type is doing the job. But that argue against some synergy between the wild type and the mutant that would somehow increase the activity. We confirm this using a knock-in mouse that contains the EZH mutation in our patient. We generated a mouse, the homozygous is lethal, but the heterozygous mouse is healthy and even shows some overgrowth. But importantly, the mouse allow us to look at EZH activity in vivo. So by staining H3K27 methylation in embryos, we were able to show that in both homozygous and heterozygous mutants, there's a decrease, but not complete loss of EZH activity. Therefore confirming in vivo that EZH2 mutation reverse indeed a partial loss of function. Now, if it's really a partial but not complete loss of function, then we have to ask the question, why is that? How about a complete loss of EZH2? Why doesn't that more commonly cause Weaver syndrome? And our working hypothesis to that question is that maybe overgrowth depends on the overall EZH2 activity. So for example, in a normal person, you could have 100% EZH activity, and that gives you normal growth. In a heterozygous Weaver mutation, maybe you're down to like 75% activity, and that leads to the repression of some genes in the growth plate, and presumably many of these are growth hormone genes, and so you get a little extra growth. But when you're down to 50% in a heterozygous null mutation, the repression of more genes happens, and so maybe you get other problems. It just doesn't show up as overgrowth or Weaver syndrome. Now, whether that's true in the other two overgrowth syndrome, it's unknown, especially because, for example, in SOTOs, there's more loss of function mutation found that cause SOTO. So we don't know if that's true in the human. But in mouse, there's some similarities. There's the NSD1 and the DNMT3A knockout mouse that are both small. And the only mouse model that gives overgrowth is a heterozygous point mutation of DNMT3A, which is kind of similar to our Weaver syndrome. So we'll see if this titration is true in the other disease too. And then there's one last interesting mystery about this epigenetics connection with the overgrowth. It's why there are 3 or perhaps more overgrowth syndrome that are caused by mutations that affects different epigenetic marks. Could there be some underlying mechanism that would tie them together? And I'm just going to spend the last couple of minutes to mention some, a couple of recent studies that might have provided some insight. There's this 2019 Nature paper that found connection between previous NSD1 and DNMT3A binding. So it turns out DNMT3A has a PWP domain that can recognize and bind to H3K36 dimethylation. And in this study, using some embryonic stem cell, when they do ChIP-seq, they found that H3K36 methylation and DNMT3A were both co-localized in some intergenic regions outside the gene body. And then, but that could be coincident, But then when they use NSD1 knockdown, which is a little similar to SOTO syndrome, what they found is there's a loss of H3K36 methylation, which is what you expect because NSD1 control this. But interestingly, they also found that DNMT3A is no longer binding to this intergenic region. They were being redistributed into the gene body. And so that would suggest that DNMT3A binding is actually dependent on H3K36 methylation. And so perhaps in SOTO syndrome or Tedder-Moran syndrome, whether it's caused by NSD1 mutation or DNMT3A mutation, they could both involve an intergenic recruitment, loss of intergenic recruitment of DNMT3A, and that would in turn lead to hypomethylation of these intergenic region in the chromosome. And then there's a second study, which I'm not going to spend time on, that show similarly the NSD1 control H3K36 methylation can affect PRC2 complex mediated H3K27 methylation. So maybe H3K27 is also dependent on NSD1 activity. And so there you have it. Maybe all 3 overgrowth syndrome could share a common pathophysiology. Maybe it's all dependent on H3K36 methylation controlled by NSD1, which in turn affect the genome distribution of DNMT3A or H3K27 methylation. Now, how these changes in epigenetics internally to overgrowth and how that affects the growth plate is another question, and I hope one day I'll have an answer for that. But I think I just ran out of time, and I'm going to show my acknowledgement, and then I was told that next slide would just have some question magically show up.
All right.
Maybe not right now. That's all right.
Well, Julian, thank you for a beautiful talk. So first, we're going to invite people up to the microphones in the room if you have questions, and I have some questions myself, but it looks like we have Dr. Cutler has a question.
Gordon Cutler, Deltaville, lovely talk, real exciting. As I listened to this about an April paper in JCNM from China, Shanghai, with 111 different genes that resulted in severe short stature that looked genetic. And they ended up finding in about 800 cases, 45% of them with these structural mutations. And I'm wondering about the other 55%, and this is speculative, but do you think we're missing in many other genes, epigenetic or regulatory changes that don't show up in the structural gene, but are working through the same pathway, but through an epigenetic or the regulatory DNA?
Now, these are short stature, right?
Yeah, these children had short stature, and they had one of 10 other features, like maybe a little skeletal dysplasia, a little facial morphism, things like that that suggested a genetic possibility.
That's certainly very possible. As I mentioned in some of my slide, there's a whole big list of genes that was found to be epigenetic regulators, and I've only talked about very few. And there are a lot of these genes that haven't yet been connected directly to growth disorders. So far, genes that cause short stature on that list mainly affect H3K4 methylation, which makes you think that there could be more, because there's actually a lot of genes that could affect H3K4 methylation. And because H3K4 methylation can also be connected to histone acetylation, and there's a whole long list of genes that's controlling acetylation, so very, very possible.
And what about, this is, again, not directly related, but it just seems to me there ought to be mutations in the non-coding DNA that are important regulatory, not so much opening up the histone, but just in the regulatory itself. Are there just not that many? Is that just something that for, survival or other reasons just doesn't happen very often?
The question is, what is the...
Well, it's sequence mutations in the promoter or in the binding sites for transcription factors or in the non-coding DNA. It just seems like there ought to be a lot of mutations causing growth failure that way, but maybe I'm wrong. Maybe there are lots that I just haven't come across them. But
well, I would think, because a lot of the current technology focus on the exome sequencing, and so we're missing a lot of possible mutations that were elsewhere. And I think, for example, there's like a recent paper in Cell that found like mutations in like enhancer regions that could cause a disease. So we could potentially be missing a lot of things that we're not looking.
Thank you.
So Julian, I have a question for you. You beautifully laid out how there are essentially monogenic disorders that lead to significant perturbations in these methylation, leading to these either overgrowth or undergrowth syndromes. So I guess my question for you is, do you think that there is more subtle variation in these epigenetic, marks and modulations that play a larger role in regulating height in the general population and in short stature? And like, where are we in evaluating the child who comes in with short stature and saying, let's look at your epigenome and how that plays a role.
So I think there are two questions here. The first is, on top of these mutations, whether you can more subtly affect height by these genes. And the answer is definitely yes, because what I didn't mention is just, for example, EZH2, it was on the GWAS list that, maybe Joe is going to tell you more. EZH2, it's one of the genes that was found to have a polymorphism that could affect very subtly how the height in the general population. So I didn't look at the full list and compare to all the genes there, but obviously you could have polymorphism that is in these genes that could more subtly affect height. And then the second question is...
so I guess, though, we're still looking at
like epigenome.
Right, we're still looking at the markers in the genes as opposed to looking at the methylation marks themselves, as opposed to looking at the epigenome.
So in terms of looking at the epigenome, I think the people who are studying DNMT3A is trying to do something which I think is very interesting. They were trying to look at how DNMT3A mutation could affect like DNA methylation in the blood cells, and that was done only in the mouse. But let's say if they found something interesting, you could more broadly screen patients with DNA methylation, and maybe that you can use it as a screen. But in terms of histone methylation, I think that's a more complicated system. And because right now we don't have a full understanding of how these methylation actually affect the growth plate, I think we're not there yet, but maybe in the future.
Brad Miller from Minnesota. I know there are mouse models where you can generate intrauterine growth retardation, and I'm wondering if any of these epigenetic tests have then looked at what happens in the growth plates to genes regulating this area or epigenetic changes, and can that then be applied to children with SGA and poor growth with growth hormone resistance phenotypes?
Yeah, I think that's a very interesting area to look into. I think, unfortunately, a lot of the mouse model that was available, I look in the literature, there isn't that much work on that specifically on the growth plate. And we are one of the group that has looked more carefully at the growth plate. But even so, our understanding is more in depth in the knockout. Even, for example, in the Weaver mouse that we show some overgrowth, it is very difficult to look at epigenetic changes that is very subtle, that is perhaps affecting only like a 25% activity. And I think we'll need some technological advance in order to make more progress in that area.
Thank you.
And perhaps I have one more question for you, okay, Julian. So I want to hear your thoughts on the therapeutic implications of this. Do you think that we'll be able to target methylation, DNA, histomethylation, DNA methylation as a potential therapeutic? And noting that these syndromes have developmental abnormalities associated with them, that there are cancer implications, how do you think we're going to be able to modulate that?
I actually thought about that. Like I mentioned, let's say the EZH activity. When you have 100% activity, you just get normal growth. But if you have 75% activity, maybe you get some overgrowth. So maybe you can, very crazy idea, use some small molecule to partially block EZH activity, and then maybe you get enhanced growth. But I'm not sure if parents would be ready to use these molecules. I think maybe it's a little too scary. So, I think that's a very interesting possibility, but I'm not sure if you can actually go to the clinic and do this.